IVT's Validation Week is going virtual for 2020 this October. Not to be confused with a single presentation webinar or Zoom training, Val Week 2020 brings a completely virtual 3-day conference experience. With nearly 30 expert speakers and a combination of live and on-demand sessions crafted with your time in mind, the caliber of training content is unrivaled. Hear from top pharma, biotech, and medical device subject matter experts, gain exclusive insights from FDA speakers, build up your team's validation toolkit with take-home checklists and materials, and leverage unlimited networking opportunities with colleagues and key decision makers. Plus, claim up to 15 credit hours toward GMP training requirements. You won't want to miss it. Visit www.informaconnect.com/validation week for all details on agenda and registration. Once again, that's www.informaconnect.com/validation-week for all event details.
This is the United Podcast Network.
This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacey.
Thank you. Welcome, everybody, to another episode of Voices in Validation, brought to you by the IVT Network. Each week, we bring you timely interviews with life science experts and discussions focused on trending topics, frequently asked questions, and industry best practices. This week, we are pleased to welcome back Igor Gorsky. He's Senior Consultant with Val Source, and he's here to talk to us today about science and risk-based cleaning validation. Thank you so much for joining us, Igor.
Well, thank you for inviting me again. It's been great last time, and hopefully we'll have fun this time. I'm happy about the subject. As you will see, we'll talk about why I'm happy about the subject. But thank you so much.
No, it's a pleasure to have you here. Yeah, absolutely. And for our listeners, historically, as you know, Igor, the pharmaceutical industry has mainly approached cleaning validation as a compliance exercise, closely linked to traditional process validation methods. And the problem with this approach is that they did not take into account science-based master plans or risk assessments. And so over the last couple of decades, the industry and the regulators have been moving toward this much more science, risk, and statistics-based approach for cleaning validation. And that's why, we're here talking about this today. So with that being said, can you provide an overview of the existing regulations and current guidances as it applies to cleaning, just to kick us off this morning?
Sure. So what I'd like to do is I want to start with the actual requirement for cleaning. And interestingly, as what you just said, the people did not take into consideration, a validation master plan, a designing of the processes, looking at the processes with regards to how we're going to evaluate the processes. And as we look at the actual regulation, so taking as an example, a good manufacturing practice, 21 CFR 21167. It basically has 3 parts. So for cleaning, and it's called equipment cleaning and maintenance. So as you see already, the cleaning is linked to maintenance rather than to process, right?
Right.
And so that's a start. So even before we go, and I may repeat this several times, usually when I do these talks, I shock people by saying that no one cares about cleaning. Okay. No one cares about cleaning. We don't care about cleaning at home. We don't care about, of course, some of us do, but most of us don't take that seriously. And obviously it's an issue. Okay.
Until there's warning letters related to cleaning.
exactly. So the good example would be I typically go to a couple of times a year. Obviously, we're in the pandemic right now, but I go to India and China, and I train people there, and they have a lot of issues with cleaning. And when I go in front of 50 to 100, 150 people, I say, do you care about, is cleaning important? And everybody's like nodding their heads. Yeah, it's very important. And I said, why, if it's so important, why we have so many observations, right?
Right.
And that's the big problem. The problem with cleaning is that you can always reclean, okay, unless you have an issue, okay, and that's obviously not a validated state. If you
right.
if you have a do-over, okay, so that's unfortunately the big issue, and it starts with a regulation, and the regulation has at least U.S. regulation has 3 steps. Step 1, you need clean equipment. I'm just going to, I'm not going to go through
absolutely
all of it, but just of it. You need to have a written procedure. Right, and the third one, you need to have an evidence that you cleaned, and that evidence, interestingly, written, so it's ABC, and that evidence is written as you need an evidence for, or you need a documentation, records kept for maintenance, cleaning, sanitizing, and inspection, in that order. Okay, so maintenance is viewed more important historically. I like that to say historically. Some people like traditionally. I hate that word with regards to cleaning because that kind of gives you an idea that you can continue with that tradition. Okay, well, not so much. Okay,
right.
So the regulation 21 CFR 21167 covers these basic points of what needs to happen for you with cleaning. Now, it's different from the process in the way that when you look at the similar regulations for the process, it actually says that the process has to be designed. Okay, here they don't tell you that this needs to be designed. They just say you have to keep it clean. Right. And this is where all of this is implied. Obviously, you can't have a chapter, 5 pages or 2 pages or a book on cleaning in CFR. But you need to have a method, and it needs to make sense. And more importantly, you need to need to measure that. You need to understand that. So when you sit in front of the regulator, and regulator asks you, how's your cleaning? And you say, good. Well, what does that mean?
It's good.
I always pass.
Yeah.
And that's an issue. That's an issue because that's the other thing that I always say. You're not good because you passed.
Right.
Okay, because you can pass at the top or at the bottom, and don't see how you trend and how you shift, right? And so the other thing that I always say is is cleaning is cleaning as important as a process? And people say, yeah, it's important as a process. I said, well, is cleaning valuable? And everybody nodded, yeah, it's valuable. Cleaning is not valuable. Cleaning is a liability, okay? Because it's time that you spend not making the product.
Right.
That's why you cannot afford to have a process that you need to repeat, okay? And not understand it, because how do I know if I'm repeating it? How do I know I didn't miss something before, right? So it starts with a regulation, and we have two major regulations. One is the FDA Code of Federal Regulations, Good Manufacturing Practice, and in Europe it is Udrelex, Chapter 4, and that Europeans learn from us, so they actually have it in a directive, and they have specifics on how to clean. It's much longer. It has it tells you that the process needs to be designed. So they kind of learn from us. Okay. So these are the major regulations with regards to cleaning. Now, with regards to cleaning validation, the only regulation that exists in U.S. right now is FDA guidance for inspection of cleaning processes. All right, and that was written back in 1993.
So it's got some years on it.
So it's been actually written in July of 1993, same year and same month as my son was born. So he didn't just went to kindergarten and high school or elementary school and high school and college. He actually got an attorney degree, and he's practicing already.
Great.
So this is how old this thing is, right?
Right.
So from the birth of my son to him being a successful lawyer. Okay?
Congrats. That's great.
Thank you. So it's very old.
Yeah.
It's very old.
Sure. Yeah, it's definitely got some age on it for sure.
Yep.
And yet we've still seen a shift over the last last 10 or 20 years to a much more, I guess, science-based, statistics-based, so evidence-based, let's say, approach. So talk a little bit more about that and the role of the American Society for Testing and Materials, what role they've had in this shift, this mindset shift.
Right. So I'll start with a little history here, and the history goes back to the age of dinosaurs. Back in 1970s, the late 1970s, there were a couple of issues with cross-contamination and a couple of issues with contamination of the parenteral products. And FDA basically said, people, you don't know your processes, you didn't design them well, so you need to use something. You need to use this word. What is that word we need to use? Validation. And so the word validation was born in about mid to late 70s. So it took them about good 8 or 9 years just to get to the guidance for process validation from those instances of parenteral cross-contaminations. And then in 1988, there was a big event where, and it was big because there were two companies involved. They don't exist anymore. Like all of these companies that existed in the past. So there was a big companies, Roman Moss and Warner Lambert, and basically Roman Moss had contamination. They were making not just a cholesterol product, but they also making a product for a pesticide. Okay. And to save on cleaning, they used the same drums as they used for the pesticides for the cholesterol product. So when they gave product or they sent product to Warner Lambert, for whom they were making that product, and Warner Lambert tested it, they've seen some peaks that were not supposed to be there.
Right.
And so they figured out that this was pesticide in there. And they figured out that the procedure was wrong, and there was a possibility, not that they were not cleaning the drums, they were cleaning the drums, but there was just not enough to clean them. Okay.
Right.
So this is where the situation started, and FDA came up then with not the guidance for validation of the cleaning process, but the guidance for inspectors to inspect cleaning processes. So that gives you like this level of importance for cleaning.
Right.
So, but there was a push, and that guidance brought in some good points, and those were basically the equipment needs to be designed to be cleaned. The cleaning process has to be written, not designed, but at least written. There needs to be analytical methods when you can test. The sampling can be done. The first time that you see swabbing, rinse sampling, routine and process controls, and establishing limits. Okay. Now, FDA did not tell you, and specifically said that we're not telling you what kind of limits you need to have. And then the industry is perplexed, what kind of limits we need to put in there. Somebody writes an article. There's this pivotal article by this guy Mullen, Foreman and Mullen from Merck, and they said, okay, 10 ppm sounds like a good limit, and 1/1000 of a dose sounds like a good limit. So at the time, as I said, nobody cares about and it seems like a low enough number. And so this 1/1000 of a dose was born, and 10 ppm was born. Where it comes from, who knows? This one article, and from then we continue having issues still. So PDA, about 5 years later, so it took them from 93 to 98, have written this guidance, PDA Technical Report 29. And it was issued 98, I'm sorry, and eventually was rewritten in 2012. And now I'm a leader of the team who's rewriting it again for the risk-based and science-based approach. So industry kind of took initiative here to give some guidance of what do we need for this cleaning business, but there was still no good regulatory guidance. You just, you knew you need to validate. You need to have some limits. Do these limits actually good or bad or ugly, or this is 1/1000? So a lot of people, as we see in the future, a lot of people were failing these limits. Why they were failing these limits? Maybe they were not rightly said. Maybe it's impossible for you to clean, because right now, as you know, the doses are going down, right? Mm-hmm. So we don't want to give people 100 mgs of something. We want to give people 50 micrograms. Right. So think about that. Now you got to clean to the nanogram, but can you see nanogram? So when you clean, right? Can't you even determine is you clean, right? Are you clean?
Sure. Right. So, absolutely.
So with that said, now industry and regulators start understanding that there needs to be more to that. We need to understand more, how to set these limits, and this limit setting kind of drove this science and risk-based approach. So once again, the industry had written another guidance, this time by SPE, a risk map approach, which is a risk-based manufacturing for pharma products, where they introduced this word acceptable daily exposure. So as similarly as we do or we measure occupational exposure, we can measure, the exposure of these materials on the surface, right? And possibility of these materials to contaminate next product. So that was one thing, and then after that, now EMEA took a leadership, and they came up with the actual now annex, or actually the guidance for health-based exposure limits.
Okay.
And that guidance said basically you need to now this 1/1000 business, 10 ppm business, old stuff. Don't worry about it anymore, because they're arbitrary limits. We don't really know if 10 ppm is good for this product or 1/1000 is good for this product. So you really need to look at toxicity of these materials, and so that started in 2012, and then The latest one came up in 2018, which was a questions and answers for that guidance, which is a very, very good document now. We went back and forward with EMEA with commenting on it, and now it has some good points. And then, our team was born back in 2012, and it consists of basically experts from a big chunk of the world. We got Germany, we got France, we got Denmark, Japan, Malaysia, Great Britain, obviously United States. So we got a good cross section of the world, and we started working on this with ASTM, which is American Society for Standards, on this risk and science-based approach, similar to the process validation, because cleaning is a process. Okay,
right.
And if we are to take it seriously, we got to look at it same as we would look at the process from perspective of ICH Q9. And so, with that said, what are these? How do these ASTM? How do they relate to the regulators? Okay, so back in 1995. There was a White House memoranda issued to the governmental agencies, including FDA, that they should be using ASTM standards because these are consensus standards. So this is not like, this team of people, 10 people got together, they wrote something, and then, everybody say yay, that's, released and approved. No, the ASTM has an E55 committee, which has who's who, not only in the industry but also in the regulatory field. So FDA is part of the balloting bodies that ballot on these standards, and then by that memoranda or by the directive from the White House, executive branch of the government, the regulatory agencies must, not should or can, must take that into consideration. Okay. Now, they don't need to tell you to follow them, but a good example would be a process validation guidance from FDA, which references about 6 or 7 ASTM. So that tells you that they, not these ones, but and we'll get to where this is referenced. So it took us a while to write this ASTM for risk-based and science-based approach, which was E3106, now in the -18, which means that it was issued in 2018. But it was balloted by FDA. We did spend time with FDA talking about it, and they're favorably looking at it. Now, the interesting part is that now it start. The regulators now start looking at it as the real thing. This is what we need to do. And so instead of our historical approaches where we basically went clean and failed, okay, and recleaned, now we want to see how well do you clean, okay? And that takes, we will talk about that, I think, in the future. But interestingly, in the last 6 months, WHO, who took now baton on trying to give even a better definition of cleaning validation, they now referencing this approach, which started in E3106, and then we also teamed up with the team of the toxicologists, who's who in toxicology, literally, on that team. You have David Dolan, you have Joel Burku, you got Esther Lofson. So basically, you got the entire world of toxicology present there, and in the matter of just few, 3-4 years, we wrote another ASTM E3219, which is a fairly large document on how do you set. So remember what I was talking about, the EMEA guidance on the setting limits. That was basically said you got to set the limit, and here's the formula on how to set it. This one actually tells you what is in that formula and how do you determine, and what are the examples of how you set these limits. So it gives you a guidance of how to do that. So now we have good tools, and this is where the ASTM takes takes place, and how it fits into guidances. So FDA and EMEA, obviously WHO now is embracing it. So that's how the guidances work. There are also few guidances out there in the world, to mention, the ANVISA Brazilian guidance, the Indian Schedule M, the Health Canada. They're antiquated, and I think in the next couple of years we will see more of the move towards the health-based limits and the science. And then the science is, as you mentioned, basically uses statistics.
Right.
So you measure, right? So I think that should cover that question.
Yeah, no, I think that's a huge overview. It seems like there's a lot of players in the game now, and the majority of the regulators are really not only understanding that cleaning is necessary for patient safety and for product quality, but now they've provided some tools for people, right? Sort of a roadmap to follow, if you will. And that brings us to today. Obviously, they're going to continue to refine some of those regulations over time, but they're definitely more risk-based in their approach, and that's how the entire industry is kind of moving. So, everything's starting to line up well. So like many other areas of pharmaceutical, cleaning validation is moving in this direction. And I'm wondering about the cleaning processes. Would being weighed against the risk factors and patient protection. So what are the corresponding changes then to quality risk management processes? Right.
So the when people will see, and it's a great question. So when people, when we issue, when we publish this PDA technical report 29, and we change that, the report is right now has a title of Points to Consider for Cleaning Validation. We're changing that title to Science and Risk-Based Approach to Cleaning Validation. So enough to the points. I mean, we should, by 22 years is enough to get to the now we got to the point. Right?
You think 22 years isn't long enough?
Right. Well, this is interesting. It's a very interesting actually subject, because we see now when it took FDA years to come up with some guidances, now the guidance is coming out like every week.
Right. Right. Yeah.
Now they're dime a dozen. Now, is it right or wrong? That's a different issue, but at least they're writing like on a daily basis. I think it's a good dynamic. I think it's too much. You want to really bring science into it, but I'm not going to comment on that. So when we look at the risk-based approach, right? And the risk-based approach is outlined in the ICH Q9.
Right, and we'll link to that in the show notes for all of our listeners. We'll have links to some of these guidances and some of the documents you're referring to. Yes,
and so when we look at that, this approach, and by the way, the risk assessment is not a risk management, right? Mm-hmm. So the risk assessment is a one-time exercise, and risk management is a cycle.
Ongoing. Right.
Ongoing. And what does it mean? It means number one, that you are following the idea of or the path, the work process of the risk management, right? So you identify the risk, right? In the risk assessment, you identified, you analyze it, right? you evaluate it. So analysis and evaluation, two different things, right? Analysis is, 2 plus 2 equals 4, right? And the evaluation is for what I'm looking for, right?
Right.
Maybe I was looking for 5, right? So that's a different story, right? So that's a risk assessment. Then there is risk control. That's a second kind of a bucket or second step, where I'm really looking for two things. Either I reduce the risk or I accept the risk, right? So with regards to cleaning, we'll talk about that. And then at the end, there is the output of that, right? Which is a risk review, right? So we did all of that. We evaluated, we analyzed it. We identified, analyzed, evaluated, we mitigated or accepted. Now, did it work?
Right.
Is the process working? And then you come back to it because you maintain something, whatever that is, process in this case. Now, with regards to so that process is very similar for cleaning, right? So when we talk about identification of risk, and we didn't say what the risk was. So risk are basically two things, right? Risk equals hazard. Okay, and hazard in cleaning is what? Cross-contamination.
Contamination, right?
Right. So even if we dig further, we will see the hazard is not just cross-contamination, but the level of that cross-contamination with regards to the patient. Okay, so is that material toxic? How toxic is it? Right. So our risk identification would be finding out toxicity of that material. Okay.
Yeah, absolutely.
That you're cleaning.
Yeah.
Right. So as I said, so the risk or the hazard has two components. Okay. One is severity on that side, from this side, because we're in mirror. So one is severity, and the other one is occurrence. Okay. How often does it happen?
Right.
So from severity perspective, as I said, we determine what the toxicity of material is, how much of that material we're putting in, right? We also can look at the microbiological hazard of that material. We can look at the equipment hazards. Is equipment actually designed well to clean? Now, there's a good point here to say that it's a huge misconception that the manufacturers of equipment are building that equipment for you, for our listeners and viewers. They don't care. They make that equipment to make money. That's their main. That's the main reason why they make that equipment. Okay, it's up to you to understand how you use that equipment for your process. Now, same with cleaning. You could use well. It could be great. You could use this encapsulator excellently. It'll be the best, the finest. But then you're not able to clean and you contaminate. Who cares, right?
Right.
So equipment hazard, right? And then procedural hazards, right? So some people have a procedure. Number one, equipment dirty. Obliterate all the previous stuff. Number two, clean. Number three, check cleaning. That's it.
Right.
Okay. Not that's right. It's not right because it even by CFR 21, that was the reason why I said that, because 21 CFR 21167 says specifically you need to have instruction on how to disassemble and how reassemble. Okay?
Right. And so it's important to have these processes in place. And it sounds like equally as important to train your cleaning validation team on the processes and make sure that everybody's doing it the right way every time.
Absolutely. Yeah. So those are the hazards. Okay. And then how do we determine what the other part is, right? Which is the occurrence. It's how well you clean.
Right.
This is where your statistics come in. This is when you test, when you get the results, you evaluate these results using statistical methods. Okay, And then the analysis of that would be, selection of your method, risk analysis, right? Selection of your method, development of your cleaning process, using at your previous cleaning experiences, right? And then see if you need any modifications to the equipment, like I talked about. So this is the analysis. How do you now apply that knowledge that we identified to this cleaning process? Okay, so you did that analysis. You evaluated. Now you have a procedure. You create a master plan during your evaluation process, and then you would do your qualification runs. Okay, and then after that. Then it's a little bit different from the process, because you determine the controls, or one can say that the controls are in the part where you develop the procedure. But actually, your controls are checked when you are already cleaned and you understood that the cleaning was good, because you can't really in the cleaning. Some people do some development work on a big scale, but most of the people don't. So I totally encourage everyone to do that. But that's your process, and then obviously when you calculate your capability of your process, you get to the review, and that could then be part of your quality system, where you review that with your quality folks. Okay, so that's kind of in the same way as the structure of the risk management for the processes. We applying the cleaning process, or we're applying the ICH-Q9 to the cleaning process.
Right, and it makes total sense. So you're designing the process, you're training on it, making sure your team's following the processes, and then you're reviewing, testing to make sure that everything went the way you expected it to go. Right. And that makes total sense.
And based on that, based on a hazard, right?
Right.
How bad the hazard is. So the hazard could be very bad, right? So, but you could be cleaning that very well. So why not? And so it is not how bad it is. It's the understanding of what that is and how well we can.
Right. So it sounds like, obviously, if you know that you have a high risk situation or hazard, then that gets priority in terms of how well you're cleaning, how long you're taking to clean, the whatever, the methods you're using to clean. So it's just like in the rest of the industry, right? Where we weigh our risks against our activities. So that makes total sense. So it sounds like there's a lot to developing the process and making sure that everybody's understanding it. Aside from that, Igor, can you help us understand a little bit more? Walk us through the steps for cleaning risk management processes under ASTM guidelines, and for our listeners, if you can highlight some of the tools that can make this process easier and more consistent.
Yes. So as we said in the other segment, we talked about the understanding of the product, right? Of the material that you clean, right. Understanding of your process, understanding of your equipment, and understanding of we didn't mention cleaning agents, but that's another part. Understanding of cleaning agents is agents, and when we look at the ID, for instance, what are the tools to identify? Number one, the determination of the hazards. So you probably want to not probably, based on the E3219, you must have a credible or a certified toxicologist who would determine the hazard.
Right.
Okay, who would determine the this health-based limit, right? Then you want to look at your history. Okay, so that requires you to have retrospective reviews and understanding of the products that you're cleaning, the equipment that you're cleaning, and the results that you get from cleaning. Okay, so for instance, you may have immediate release product that is very easy to clean, or you may have extended release product which has some polymers in there, and it's very hard to remove, right? Because they're by definition they're thickeners. They're like glue, right?
Right. Okay.
Right? So you need to understand that history. So what do you do for that? And that's important. That's a very important part. Cleanability studies. Okay, people bring the products in and they just clean. Okay, that's all they do. And then if it doesn't work, it doesn't work. So cleanability studies are important. And just what we talked about, that the this quality by design kind of a thing is emerging and being used in the last 10, 15 years. Same thing, right?
Right.
The other tool is visual inspections, right? Visual inspection or visual limit studies. How far can we see? What you're actually seeing with regards to the product that you are cleaning. So these are kind of spiking studies when you can look at the surface and you can see the level of what you actually can see to which level, right? And then the methods, development of your methods. Not just I'm going to use one method one fits all, but specifically for your specific application. So the basic outcome of the risk identification, is that risk identification is different for every product. If you consider every product, okay, as at least a different entity, okay,
right,
even if it's very similar, and you take time to at least do a risk assessment, maybe it's all the same, and you do the risk assessment and say, hey, it's all, yes, this fits, this fits, this fits, we don't need to do this, we don't need to do this, okay, but at least you are giving it a chance to go through that checklist of these tools. Do I really know how much would be left on the equipment, and can the operator see it when they clean it? Okay, so if I have a 50 microgram product, right, I can't really have a 5 nanogram as a limit because I can't really see it.
Right.
And by the way, the lowest level that people typically can see is probably somewhere around 0.5 micrograms. So people can see far below. And then for the risk controls, we're talking about the cleaning validation studies. So these are the statistical methods. And by far, the most important one is the capability analysis. So it's calculation for your capability indices, CPKs or PPKs. And then you can also compare different runs, do sample t-tests or some other statistical tests that that could be used. Okay, so
Sure.
a lot of these, and we'll provide a link to some of these articles. There's actually a very nice book that we compiled with our articles, where we can actually not that we're advertising it because it's free.
No, but we can link to it. It is a great resource. It is a good resource, Igor. Right. So we will link to that in the show notes as well.
Right. And then the other tool that I definitely promote for people is to use this for information communication based on information communication basis. So report that in quality forums, quality councils. Report that as a quality metric. Okay. Because it's as important. How well you clean is extremely important. And when I go to the companies and I audit them and they say, well, how well you clean? Well, we have some failures. What are the sum? Right.
Right.
Is the sum a lot or not a lot?
what's the impact of it, right? And
right. People typically re-clean. That would be the impact, right? But we'll get to what the impact is. Yeah, I hope that answers.
So thank you. No, and so it sounds like there are some tools out there that people can make use of. And would you say, because we're talking about high hazard and high risk situations versus lower risk, lower hazard, would you say these tools are equally valuable and necessary, I guess, for the lower risk, low hazard as they are to the high risk, high hazard?
Absolutely.
Okay.
Unless you build knowledge and the industry is struggling, I think we in the last in my last podcast we talked about the quality management system, or pharmaceutical quality management, and people here, we just talked about the risk management, right?
Right.
But people are struggling. So when you look at that ICH Q10, the quality pharmaceutical PQS, it says that it has two enablers, right? One is a risk management, and the other is knowledge management.
Right.
So unless you understand what that knowledge management is, right? So I can't decipher between the low and the high. So okay, I deciphered between the low and the high. This product has a low toxicity. This one has a high toxicity. But maybe it's hard for this product to be cleaned.
Right.
So every product is different, and unless you start understanding what the continuum is, right? Or if you disregard the low level risk, you will fail. Right. Because eventually you will have an issue. Because remember that adulteration, right, doesn't measure the risk. It just says you cannot have this substance in this product, right? Otherwise, it's not going to be pure, right? It's got to be safe, pure, and effective, right? So it's not going to be pure, right? So it doesn't really matter if the level is low. If I'm contaminating with the low levels of contamination, who cares? It's contaminated, right? So it is the same.
Good point. That's a great point.
And the part of that, just very, very quickly, the part of that also, you've got to make sure that that you are consistent in evaluating everything the same way. Otherwise, you're going to lose in this approach. Okay, you're going to lose your system, right? So the system should be staying the same. Okay.
Right. And so, Igor, we've talked about the processes and the methods and the tools. We know in the industry and in the world, quite honestly, things are rapidly changing. New technologies are being developed and embraced all the time. And so I'm wondering if these techniques and methods, how they're designed to be anticipatory of emerging technologies and advances in medicine, because we are moving quickly. And now that we're in this pandemic, we're moving even more quickly than we were. So
correct. So the future of cleaning, right? We're moving very fast towards single-use materials. Okay.
Right.
Single-use equipment, which is good, okay? But the hazards are still there with regards to leachables, extractables. So you're still looking at the evaluation, at least analysis of the hazard. Okay, so the very similar approach. We're not talking about cleaning it now. We're talking about cross-contaminating with something that comes with individual use products. Okay. The other part is you're still going to be looking, even if it's not individually, even if you share the facilities, you're still going to be you need to understand what do you contaminate, or what are you cleaning, and if you are sharing, what is the possibility or what is the probability of contaminating? right?
Right.
So that all of that will still be the same. You will still need to evaluate. It doesn't really matter if it's what kind of technology is out there, but you will still need to measure your cleaning process. Okay, so the statistics and the hazard evaluation will still be there, regardless. That's the beauty of the system, that it provides you with a tool to evaluate. Okay, before you just go and clean. Now you know what you're cleaning, and you know how well you clean.
Right. Absolutely. So, so technology will be useful to this process, as it is to many others. So with the ultimate goal then being patient safety, as we've mentioned, throughout pharma, biotech devices, this isn't different. Patient safety is the ultimate goal. We've seen industry shifts in quality by design, and you started to mention that a little while ago, and quality systems assurance practices. So this is undoubtedly in an effort to plan for highest quality, efficiency, and continuity from the beginning. Why do you think cleaning validation needed to be removed from under process validation in order to make this happen?
Well, as I said when I started, cleaning is a process, and we are frequently fail this process. And if we don't take that, if we don't take a charge and identify it as a process, okay. Now, I actually would be happy if people would bring cleaning into their process, okay, into the cleaning, into the batch record. So a lot of companies, I advocate for a lot of companies not to finish when you finish the product, but to finish when you finish cleaning this this product, and your batch record, your cleaning is a part of that batch record. It's especially advantageous for manufacturers of vaccines, for manufacturers of gene products, right? Because you don't need to let these materials, let these proteins sit there. And as we know, the proteins are sticky materials. A lot of them are sticky materials, so it's hard to get rid of them. Okay, and it could actually kill your next product. Not necessarily even contaminate, but kill your product, right?
Right.
So that kind of my point to that is that take it seriously, take it as a part of your process, okay? But take it as a separate part and as important part as as a part of making the product.
So with all of this in mind then, Igor, we've talked about a lot today. And for our listeners then, what are the first steps necessary for them to take in creating a streamlined cleaning program that really ensures patient safety and product quality?
So number one, you must understand your products. So you must understand the hazards and understand the health-based limits for every product you manufacture, for every excipient you use, for every cleaning agent you use, because these hazards are the ones who could potentially contaminate. And
right.
You need to know all these numbers, okay? The acceptable daily exposures. Number two, you need to understand how well you clean. So you need to you need to calculate your cleaning results. You need to calculate your capabilities. You need to understand, how well you clean. And it will tell you which equipment is bad or hard to clean. It will tell you which products, and once you have the numbers, the math will tell you. Okay?
Right.
So
sure.
these are the main steps, and then use the risk assessment. Okay, you bring this knowledge into the risk assessment and understand if as we talked about the hazards with regards to the equipment, the hazard with regards to the microbiological hazard. We didn't talk a lot about it, but it should be there. The procedural hazards, meaning we didn't talk much about the procedures. I'm a huge advocate for the very detailed records, which allow you to learn from. So if you do have issues with cleaning, you record that. Okay, you don't put it under the rug. You record that, so you improve on your cleaning method. Okay.
Sure.
So people afraid to fail. Okay. So those are the major steps. And then if you have a legacy program, you need to evaluate all of that, what I just said, for legacy products. Okay?
Of course. Well, thank you so much. That's excellent. And it gives people something that they can do today. They can start with step one and move forward. I appreciate all of your knowledge and sharing it with us. And as we wrap up today, Igor, I want to see if there's any last, tidbits or tips that you would like to leave our listeners with.
Yes, and I kept that. So I wrote an article about 5-6 years ago where I talk about the people that clean, right? And so when we clean and we fail, we blame people that clean. But typically they don't design the cleaning, right? They don't validate it, right? They don't test it. They don't sample it. So why are we blaming, right? So that's what important to understand that cleaning is not a chore. In pharmaceutical industry, it is not a chore. Okay, it is a process. Okay, so it's nice to call it a chore, but I want that chore to be done. Not like, my son cleaning dishes after pizza, right? He's going to sit there for 2 hours.
Right.
No, you want to do it, and you want to understand what you're doing, and you want to measure it. So those are last words, Cleaning is not a chore. Cleaning is a process. Cleaning is important not because I said it's important, okay? And acknowledge that it's a liability, but if we understand that liability, we can reduce the liability, okay? All right?
That's the most important thing. Yeah. Thank you. We're going to leave it with that, Igor. I really do appreciate it.
Sure. Thank you.
It's been great to have you here today.
Thank you for inviting me. I hope it was interesting. I hope people will like it, and I'll send you some of these links.
Yeah, perfect, and we'll include them in the show notes.
Yep.
Great.
Yep.
Thank you, Igor. And a big thank you, too, to our production team, Ben Kitchen, and the entire Studio 21 Podcast Cafe staff. Lastly, a big thank you to you, our valued listeners. We really do appreciate you tuning in each week. If you have topic requests or guest suggestions, we'd love to hear from you about that. If you enjoyed this episode, we ask you to subscribe in your podcast player of choice so you never miss an episode, and share it with your friends, colleagues, and online networks. Today's show notes and information on this podcast, please visit www.ivtnetwork.com. We'll be back again next week with another great interview. Until then, stay healthy and safe.
Yep.
The views and opinions expressed by the hosts, guests, or callers of this program do not necessarily reflect the opinions of the Studio 21 Podcast Cafe, the United Podcast Network, its partners, or affiliates.
IVT's Validation Week is going virtual for 2020 this October. Not to be confused with a single presentation webinar or Zoom training, Val Week 2020 brings a completely virtual 3-day conference experience. With nearly 30 expert speakers and a combination of live and on-demand sessions crafted with your time in mind, the caliber of training content is unrivaled. Hear from top pharma, biotech, and medical device subject matter experts, gain exclusive insights from FDA speakers, build up your team's validation toolkit with take-home checklists and materials, and leverage unlimited networking opportunities with colleagues and key decision makers. Plus, claim up to 15 credit hours toward GMP training requirements. You won't want to miss it. Visit www.informaconnect.com www.informaconnect.com/validation week for all details on agenda and registration. Once again, that's www.informaconnect.com/validation-week for all event details.